Okay. So, can you see
Yes.
my screen?
Yes.
Okay, perfect. Thank you very much for your warm presentation. I'm Marta Turégano López. I'm a postdoc in the Laboratorio Cajal de Circuitos Corticales with Javier de Felipe. And I'm very happy to be here. Thank you very much for coming to my talk. I'm happy to be part of the CyberNet family and to have the opportunity to research on neurodegenerative disease. So thank you very much. And this is my presentation called Alterations of the endosomal pathway in the hippocampus of Alzheimer disease patients. So first of all, I would like to introduce what is the endosomal pathway and in particular the multivesicular bodies. Then we can explore what is the role of the endosomal pathway in Alzheimer's disease. And then I'm going to introduce you the electron serial microscopy to study the multivesicular bodies. And to finish my presentations, I would like to show you some preliminary results that we have about the alterations that we found in Alzheimer's disease samples. So let's start. Well, what is the endosomal pathway? Okay, the endosomal pathway is a system that is conserved in all the cell types. It starts with the endocytosis of a receptor, a molecule, by the invagination of a cell membrane, and then the vesicle is created. That is called early endosome. This endosome has a typical shape with tubular protrusions, and they are very related to the recycling functions in cells. So they can go to other places of the cell, fusion with the cell membranes, and insert the receptor of the proteins in other places of the cell. If the early endosomes continue in the pathway, madurating, the early endosomes transfer to a late endosome. Here, I want to clarify the term of a multivesicular body, because the multivesicular body is defined anatomically like an organelle with a simple membrane, as you can see here in this electron microscopy image, full of vesicles. And this term coincides with early endosomes and with late endosomes. So we have to be careful about that, because they are the main organelles of this pathway. When the late endosome is created, or when it's created, they go to support mitochondria along the cell, and they can release their cargo to the external medium by exosomes, by fusing the cell membrane, releasing exosomes. And it's typical too that they can fuse with another organelles from the autophagy pathway called autophagosomes, and we can find hybrid organelles with a vesicle content and a multilaminar content called amphisome. It's an hybrid organelle fused by autophagy pathway and endosomal pathway. So at the end, the target of both kind of organelles is the degradation of the content that they have in the lysosome. So for that reason, in all the pathway, the pH is going down to produce the separation and degradation of the content. So now we have this, and something that I want to highlight is that this pathway is particularly important in the neurons. This is for several reasons. One of that is that The neurons are the most asymmetric cells that we know, because we know that the synthesis, degradation, and the metabolism is in the soma. And in most of the times, the proteins that are synthesized are needed in other places very far from the soma, like dendrites, like the axonal terminals. So the neurons need to have an efficient transport system. For that reason, the endolysosomal pathway is very important. The second reason is that neurons are most of the time postmitotic. So the division in neurons is infrequently, and this means that all the components that the neurons have to be maintained along the lifetime of an organism. So they have to have strategies to recycle, to degrade the molecules that have some errors, mistakes, or something like that. So it's important to maintain the components that they support the function in a neuron. And last but not least, although the system seems to be rigid because it's no division in the neurons allow the neuroplasticity. They are a system very dynamic because they need to be plastic in the dendrites, because they need to generate new spines or to eliminate some spines. They need to be plastic in the synaptic junctions because they have to recycling the neurotransmitters and to recycling the receptors, and they have to modify the pathways and the cascades that allow the long-term potentiation and long-term depression. So the system has to be dynamic and plastic, and this is supported too by the endolysosomal pathway. So here is the key of the topic. We can see how an aberrant accumulation of proteins is found in most of neurodegenerative disease, and Alzheimer's disease is not an exception. As you probably know, there is an accumulation of tau and A-beta, and this accumulation inside of the cells, inside of the neurons, occurs inside of the endosomal compartment. As you can see here in some electron micrographs of immunogold of tau, you can see how the tau is inside of the multivesicular bodies, and how the A-beta labeled with immunogold 2 is inside of these multivesicular bodies. So the question is, are the MVBs, the multivesicular bodies, allies or enemies for the progression of the disease? Because they have a role in degradation of the proteins that are aberrant or are not working very well, but they could propagate these proteins, malforming proteins in some way, to the extracellular medium by exosomes, so they could replicate a prion-like way for this disease. So this question is not clarified yet for science, by science. So for that reason, we think that it's a good point of view to characterize in detail the multivesicular bodies that we could find in Alzheimer's disease samples and compare all these parameters that we could characterize with the multivesicular bodies that we have in control samples. The parameters that we consider that are important are the MBB's density, the density of multivesicular bodies, their size, the correlation between multivesicular bodies and synapses, because the multivesicular bodies are supporting the synaptic function, the distribution of multivesicular bodies in dendrites, axons, and non-synaptic elements, mostly glia, and the association of multivesicular bodies to mitochondria. To do that, we use serial electron microscopy in human samples. So we have 3 cases with no neurological alterations and 3 cases with characterized as Alzheimer's, as samples with Alzheimer's disease. And we study the CA1 region and the entorhinal cortex because there are regions that are very vulnerable to the disease. And as you can see, we don't have yet a good balance between male and females in both groups. For that, I check if we have difference between male and females, and in the density we don't have any difference, but we found that female MBBs are longer than those that are in samples of males. So I'm not really sure about if this result is related only by sex, because as you can see here, the males, the samples from males are with an age, minor age, so it's needed to have more samples to have a more balanced distribution of the samples. Okay. So to study the samples, we identify the region that we want to identify with a blue toluidine. And then we use the FIB-SEM microscope. I don't know if you are familiar with this microscope, but it's an electron microscope with the 2 beams. One of them is the SEM that scans the surface, and the other beam is a gallium beam, call it FIB, that mills the surface with 20 nanometers in our case. So both beams are working continuously and they can conform a stack of images that represent 3-dimensionally the tissue that we want to study. We avoid the soma of the cells because we study the neuropil where most synapses are located. As you can see here, we did trends, and here you can see the front edge of the trend. And we study in the entorhinal cortex 2 layers, the layers 2 and 3, 18 stacks of Alzheimer's samples, 18 for control samples, 974 MBBs were segmented. And in CA1, we explore 3 layers, lacunosum moleculare, radiatum, and pyramidale. We obtained 24 stacks from Alzheimer's samples and 27 from control, and 1,434 MBBs were segmented. So now I would like to introduce you Spina software, that is a software that we have developed in the laboratory to segment the objects that we can identify in our electron microscopy images. This is an example with a synapses that we have segmented, and we can obtain all the data from this segmentation, like the size, the spatial coordinates, and other tags that we can add to our segmentation. And now, in the last year, we have developed a tool to trace the fibers in the images that we have. And we can do the skeleton from dendrites, for example, that we can see in blue, that connect through the synapses to the axons. So in pink, you can see the axons, and then we can go through the connectome of the sample. This is a very useful tool to localize to see where the MBBs are localized, if they are in dendrite, axons, or in non-synaptic elements. So then we can take it every time that we want. Okay. So now I'm going to show you the images from multivesicular bodies that we have from our samples. As you can see here, this is an organelle with a simple membrane with vesicles. It's important to say that we don't use only one image to identify the multivesicular bodies. As I said before, we have an image per 20 nanometers, so the multivesicular bodies appear in more than one image, and this facilitates us to identify well the multivesicular body and don't have some mistakes or some mistake and not identify other organelles as multivesicular bodies, as here the mitochondria. So this tool helps us a lot of. And we can see here the 3D reconstructions that we have, an example, and a 3D view that provides SPINA software that is very interesting because we can see at the same time in the same sample the excitatory synapses in green, the inhibitory synapses in red, and the multivesicular bodies in yellow. Okay. So these are the first results that we have, are preliminary, but we can see some trends. We can see that the density of multivesicular bodies is decreased in Alzheimer samples. So this trend, or this result, is more extreme in entorhinal cortex. Then we measured the size of MBBs, and we found that was very similar in Alzheimer's disease and in control samples. We didn't have any difference in entorhinal cortex, but in CA1 we can see a slight difference, a little bit of the increase of size of MBBs from Alzheimer's samples, but we have to be careful of that because I have experience on this kind of results and of data, and here we have a sample very big, like 1,434 MBBs, and a slight difference could provide a statistically significant difference, but could not be so strong to be a biological difference in the system. So we have to be careful about that. And this is difference because in other models, other animals that I have studied the multivesicular bodies and this system, there is a correlation between density, an inverse correlation between density and size. And the samples where the density was decreased, the size of the MBBs are usually bigger. So this is like the first difference that I found between mice and humans. Okay. We have observed too a slight correlation of MBBs and synapses in mice, both in mice and in humans. And as you can see here, we have a correlation of 0.3, 0.6 in the control samples in both regions, and this correlation is lost in Alzheimer samples with 0.1 and 0.0. This is different that we have found. This could be due maybe because the NBVs are supporting the synaptic function because they are playing a role in the recycling of receptors or factors that are involved in synaptic plasticity too, and they support the mitochondria function that are the main supply of energy for the synapses. And I think that, here we didn't find the relation between the NBVs and the synapses in Alzheimer's, maybe because the NBVs are doing other functions that are not related to the synapses. So the system is changing in the Alzheimer's disease. And as I said before, we can identify where the multivesicular bodies are located. Here I have an example of electron microscopy images. These are from mice, but it's only to show you how the multivesicular bodies could be located in the terminal of excitatory reactions, inhibitor reactions. Here we have the multivesicular bodies. Here we can identify them in a myelinic axon, in glial cells, in dendrites, and even in the dendritic spines. So we have identified that. And then we can see that the main percent of multivesicular bodies are located in dendrites, okay? But the most different thing that we can see is that the multivesicular bodies that are located in non-synaptic elements that are mostly glial cells increase if we compare the CA1 control and Alzheimer CA1, so the sample of Alzheimer in CA1. So from the 22% to the 28%, and this trend is more extreme in entorhinal cortex, that where the 34% of multivesicular bodies are in non-synaptic fibers, and this percentage increased until the 47%. So this, I think that could be due for two factors. Like one of them is that for some reason in Alzheimer's disease, the multivesicular bodies are more frequently found and are more frequent or are increasing in number in the glial cells. That could have sense because they could do some extra degradation or some other functions that are not in the control samples. Or the other theory that I have is that because we have in Alzheimer loss of synapses, we can't identify if the multivesicular bodies are in a fiber that is receiving or is doing a synapses, if it's an axon or a dendrite. So maybe they are in fibers that are losing synapses because the Alzheimer's pathology. Okay, and now I would like to go deep in the relation between multivesicular bodies and mitochondria. This is a relation that we have explored before in other works in rat and mice. And as you can see here in this serial of images of electron microscopy, we can observe how some of the NBVs are docked directly to the mitochondria. And this is a reconstruction to the mitochondria. Sorry. This is a reconstruction, and this relation is not only physically, but is functional, because MBBs are supporting the mitochondrial function. They are degrading molecules affected by ROS, and they act as platforms for local synthesis for proteins that the mitochondria needs. So they have a function in the maintenance of the mitochondria. This is an event that we have seen too in our samples in humans. Here you can see how a multivesicular body is docked to a mitochondria. And the percentage of this association is decreased again in Alzheimer's disease. As you can see in control, we found that 24% of the MBBs are docked to mitochondria, and in Alzheimer of CA1 samples from CA1, 19% of them are docked. But in entorhinal cortex, the percent of multivesicular bodies docked to mitochondria is 21, and this percentage decreased a lot in Alzheimer's samples from entorhinal cortex until the 9% of them docked to mitochondria. So again, the more dramatic changes are seen that we have seen occurs in the entorhinal cortex. So I have some take-home message for you. This is a demonstration that electron serial microscopy is a tool that is very useful to characterize human MBBs and to compare it between samples from control and Alzheimer's disease, even with other samples from other animals. There are differences in density in the relation with synapses, the location and the association with mitochondria between Alzheimer's disease and control samples, and it seems like the basal activity of MBBs is altered in Alzheimer's disease, because we found very conserved numbers of the association with synapses and with mitochondria, and these associations are lost in Alzheimer's disease. We need to do further analysis about the MBB size because the results that we have are not conclusive. And the next step that we have to do, of course, because this is a very preliminary work, is a better balance of male and female samples and study between the layers that we have, because sure that we find difference between layer 2 and 3 from the entorhinal cortex and piramidale, radiatum, and lacunoso moleculare from CA1. We want to study the spatial distribution of the multivesicular bodies, if they are clustered in a cluster distribution or if they are randomly in our tissue. And of course, we want to go deep in the study of the relation with mitochondria, if the size of these MBBs that are supporting mitochondria is different, or if this relation is more frequently found in axons, dendrites, or glial compartments. So now I would like to invite you to this project that I have. Corpus Curiosum is a project that I have with a group of friends to stimulate critical thinking in neuroscience. We have the support of Ferns and Eyebrow, and if you are interested, you can find us in this QR. You can find all our media. And this is an example of the agenda that we had the last June. We have lectures about topics that are a little bit out of the main topics in neuroscience, and we have all these lectures in our YouTube channel. And in the last year, we create a school for students to talk and debate about some topics like neuromites, the bias from society to neuroscience and from neuroscience to society, and some philosophical topics that I think that are needed in the neuroscientists of the future. So this is all. Thank you very much for your attention. I'm here to answer all the questions and to have feedback, because I think that we can debate about this a lot. So thank you very much.
All right. Thank you very much, Marta, for a very nice, wonderful presentation. So the paper is now open for discussion. As I said earlier, if anyone is interested in posting a question to Marta, please do so by raising your hand in Teams application, or by sending me a message through the chat. I will give opportunity to everyone to
Of course. If you work with something like related to autophagy or endosomal pathway, maybe in a molecular way, I'm very happy to debate about that, to have feedback and to see what are the limitations or the other things that we can use to study this.
All right. Okay, so all right, so any questions? Yeah, we have a first question from Carlos Saura. Carlos, go ahead.
Hi, Marta.
Hello, Carlos.
Hi, how are you? Well, very extensive, very detailed analysis, morphological study of multivesicular bodies in Alzheimer's disease, and it's clear that I mean, it's sometimes it's a very painful, extensive techniques, but you have really performed a very detailed analysis of these multivesicular bodies in eddy brain that is very relevant for the disease process. I have a lot of questions, but one thing that could be interesting also is to know whether this the number, the density, or the shape of the multivesicular bodies are age dependent, because in your samples, for instance, there is a clear age differences between the controls and the Alzheimer's disease, and I understand that because you are analyzing samples with no pathology, so they are kind of younger. So do you think that, or have you seen differences in age-dependent differences in the multivesicular bodies?
Okay, this is a good question, because in fact the endosomal pathway is a very dynamic pathway. So the multivesicular bodies are all the time madurating and fusing with other vesicles. So I think that the relation between the density of the multivesicular bodies and the size is important, because I think that the system is always in balance. And if you have a lot of vesicles, you can have big multivesicular bodies, because the cell membranes has to be like constant most of the time, and they waste energy. So I think that maybe the ratio, or it's a thing that I suppose, the ratio between the size and the density could be always constant, more or less, in the systems that are in the same conditions. And this ratio, I think that is broke in altered conditions. So this is one of the things that I have surprised, because in the literature, they say that they found bigger endosomal compartments in Alzheimer's disease or neurodegenerative disease. And it's a thing that we don't found, and we we didn't found the increase of the density of MBBs. So I don't know if there is a broken balance of that, or I don't know what is the meaning of that. Or
well, it seems that you see a decrease, no, in the number, but not in the size, no. Somehow
exactly.
Indicates that
I thought that with the decrease of the number, I thought that I will have an increase of the volume. That is the thing that I found in rat and mice, in mice, and that doesn't occur.
Yeah. In terms of the source of these multivesicular bodies, no, and you have in your introduction, there was this fusion to autophagosomes or other organelles. Which is the source of these multivesicular bodies? I mean, are they more enriched maybe in autophagosome markers or mitochondrial marker? I don't know. Have you tested the nature, no, of these multivesicular bodies?
So the amphisomes, that are the hybrid organelle from autophagy and endolysosomes, you can identify, and they are different from the multivesicular bodies, because the multivesicular bodies has only the vesicles inside, and the amphisome you can see the multilaminar content. It's like the image that I put, like in the middle of the organelle is occupied by multilaminar content and the other middle is occupied by vesicles. So as we have electron serial microscopy, we can see the full organelle and you can see how the content is changing. So this work is only done in multivesicular bodies, not in amphisome organelles. The origin of these multivesicular bodies, I think that is a lot of, they have a lot of origins. Like I think because with molecular techniques, they have labeled some factor of growth, no? Mm-hmm. And receptors, tau and abeta. So a lot of, or most of the protein that you can find in the neuron, I think that in some way or some time go inside the multivesicular body, because it's that main transport for the retromer complex, trans-Golgi network, and all these paths that are conserving the function of the neuron.
Okay, all right. Okay. Thank you, Marta.
Thank you very much.
And congratulations for the work.
Thank you, Carlos.
All right, thanks, Carlos and Marta. More questions. Don't see any raised hands.
If not, I can continue.
Sure.
Just because because it was I mean, you talk about at the beginning, no, with this staining, with the characterization of the glutamatergic and inhibitory synapses, and then you didn't talk anything about, I believe, where these multivesicular bodies are more enriched in these glutamatergic inhibitory terminals. Yeah. I don't know if you have any data about that, if they are more enriched in glutamatergic or in inhibitory terminals.
Yeah, that is a good question and a difficult question, because I have the data about the percentage of the MBBs that are in inhibitory fibers and excitatory fibers. But the thing is, in the neuropil, in the regions that we have studied, the fibers of inhibitory neurons are very low. So this doesn't mean that the enrichment of the fiber. So to do that, we have to do an serological study about how much volume is occupied in our tissue for inhibitory neurons and excitatory neurons, and then we have to do a ratio between the MBVs that we have in each fiber and how is the volume occupied by this cell. This work that is very laborious because is done. I did in the rat that is published already, and in the mice that I'm publishing now, and is a good future thing to do in this kind of samples. So I will probably say to have a response of your question, but now no. But
okay, you are in the process of getting the result. Thank you.
You're welcome.
All right, thanks again. So we have another questions now from Nicolás Cano. Nicolás.
Hi. Thank you very much, Marta, for your night talk, and congratulations too. My question is, do you find any relationship between the synaptic size and the presence or the size of the multivesicular bodies regarding the synaptic, the multivesicular bodies that are associated with synaptic elements, both axon or dendrites?
Well, this is a good question. I think that the first limitation that we have is to know what MBVs are related and in that moment to the synapses, because if we do that only for the distance that where they are located to the next or the most proximal synapse, I don't know where put the limit of where I can put the limit of the distance to determine what is related to the synapse or not. This is a problem. The thing that I can say is some MBVs are in dendritic spines, so probably they are very related to the synaptic junction. So I can do the measure in that MBVs, but the thing is they are in a very low percentage of the total number of MBVs. They occupy the dendritic spine in a very low percentage. So I think that I cannot say robustly this correlation between the size and the MBV. Maybe other thing that I thought is, now that we have the skeletons, maybe we can correlate the synapses of each skeleton to the MBVs that are involved, that are inside of the cell that I traced with the skeleton. This is a good point that it could be very interesting to check.
Yeah, thank you.
Thank you very much.
All right, thank you, Nicolás and Marta again. Okay, so any more questions? Don't be shy.
Don't be shy. Some molecular research.
Somebody else? Okay, so this is last call for.
Okay, Teresa.
Okay, yeah, we have Teresa Iglesias. Teresa,
yes.
Please go ahead.
Yes, I don't know whether you hear me correctly. Yes?
Yes, perfectly.
Thank you very much, Marta. We talk quite a lot in the foro in Malaga, so
Yes.
I like to work very much, and congratulations for your talk. It's been really well presented, so that I understood better even than in Malaga with the PowerPoint. And well, just because you forced kind of the participation, that I think is very good as well, because I think we should force ourselves to participate, making questions. So mine is regarding the this decrease in the molecular the multivesicular bodies that you observe. So do you think this is due that it is like a higher speed of release of multivesicular bodies from these cells, so that the final rate that you see is a decrease, but you actually have a speedy process so that you see less then? Or what would be the explanation? Because you base your ideas on that this is going to be favoring the dissemination of the favor the dissemination of the release of aggregates to the from the cells to the brain. So
yeah, it's a very good question, because in fact we study the neuropil, and in the neuropil we can see fibers that are very far away from the soma. So it could be that all the multivesicular bodies are going in the way to the soma, and maybe are collapsing there. So this could be one of the reasons that we can see more multivesicular bodies in this part of the fibers.
So you think is favoring like a retrograde transport of
exactly
organelles to the soma, and just getting rid of them in the places where it was at synapse.
Yeah, in fact, if you have tau aggregates that affect to the axons, they can have more problems to go retrogradely, no? Yeah. So I suppose that this is one of the hypotheses that I could have for that. It could be interesting go to the soma of the cells to see if there is difference between the density of multivesicular bodies or lysosomes in both samples.
Yes, you might find something there. Okay, really nice talk, and thanks very much, Marta. We can talk anytime.
Perfect. Thank you very much, Teresa.
Bye-bye.
All right, thanks, Teresa. We still have some time for more questions, if there are any. No more questions? Okay.
Okay.
Well, if there are no more questions, then it's time to close the session here. But not before thanking again Marta for her willingness to share her work with us. Very nice presentation, Marta. Very exciting. And that's it. Thank you very much for attending as well. And I wish you a nice rest of the afternoon, rest of the day. And I hope to see you, or some of you at least, in the next last webinar of these years in a couple of weeks. So thanks, Marta. Thanks, everyone.
Thank you, everyone. Bye. See you.
Thank you, Marta. Bye-bye.
Thank you.